you will be more to. All righty, so let's go ahead and check out our classroom. Let me make sure I got the right one up. Yeah, okay, good. All right, there we go. So this week is going to be all about DNA. So I'm sure everybody here has heard the term DNA before, especially if you watch a lot of like cop shows or CSI shows or something like that. They're always all about that DNA evidence that they're looking for. And you might kind of know a little bit about it is. It's like that stuff with your genes that's inside of your body and your blood and stuff like that. And so today we're really going to get into, sort of what DNA is, what it's made out of, what are its different parts, where can you find it in the human body. So we're going to kind of introduce you guys to it. If you took class Biology A with me a couple terms ago, then you may have seen some of this stuff before, so this might be kind of a little bit of a review for you. But I always like to review this stuff before we jump in to the notes on Thursday, where we're going to be talking about how DNA makes copies of itself. It's a really complex process that I'm going to try to just give you guys the most important sort of basics that you need to know for it. But DNA copying itself is really, really important. It's how we're able to make new cells and new organisms out of ones that are already existing. Yes, Ashley, go for it.
I just wanted to know if you were going to have this whole like class on like Post-it, that way I can watch it later.
Yep.
Okay, thank you.
For sure. I'm recording it right now, and so I'll make sure to post it right after class is over. No problem.
Okay, thank you.
And just as a heads up, you can always ask at the beginning of class if you want to make sure to kind of remind me about it. But I do record every single class that we do. So every single time we come together as a group, I'll make sure to record it and post it on the Google Classroom so that anybody who misses or anybody who wants to can go back and watch it later on. But you're always free to ask. Just occasionally I almost forget and have to start recording like halfway through. But yes, we got our recording going on right now. So on Thursday, we'll go into the much more complicated process of how DNA copies itself so that we can make new cells and new organisms and new beings. But in order to get to there, we need to know a little bit more specifically about what DNA is and what it's doing inside of our bodies. So that's where today's notes come into play. So today we're going to be talking about kind of the structure of DNA. What is it built out of? The different little pieces that come together in order to make the bigger molecule. So DNA is a pretty big molecule made out of a bunch of smaller pieces put together. So we'll talk about those. And we'll talk about the specific way that the parts of the DNA link up with each other. So DNA for today. All right. So DNA actually has a really cool history. For a long time, when we were first starting to really study the human body and human cells in like the 1800s, early 1900s, scientists knew that there was this weird particle, this weird cluster of stuff that existed in human cells. It was different from like the other proteins and sugars and stuff that were found in there, but they weren't sure exactly what it did. And more specifically, they did not know what it looked like. They knew that all the cells had this stuff, but they weren't quite sure how it was organized. And so people did all sorts of different techniques to try and discover the structure of DNA. And so somebody who first gave us kind of a really clear picture of a kind of general structure for DNA was a female scientist named Rosalind Franklin in I forget exactly what it was. It would have been like late 1940s, early 1950s. She used something called X-ray diffraction, which is a very, very complicated process where basically you take some sort of material, turn it into a crystal, and then take a picture of the crystal with an X-ray in order to try and figure out the structure. It was used a lot to take pictures of proteins and stuff like that, but she did it on DNA, and she took this really iconic photograph, which suggested that DNA had this sort of X structure to it, indicating that maybe instead of just being like one big long string of material, it was two strings that were kind of wrapped around each other. So it was this discovery of hers that the DNA twisted around each other to form something called a double helix. So like if you've ever seen a like spiral staircase in like a house or TV show or movie, that's kind of what a double helix looks like. And so there's these two strands that are kind of wrapping around one another. You can't really do it with two hands, but they're like wrapping around one another. So she took this picture and kind of had this first idea of what it looked like. And then two scientists that were working in kind of a nearby lab on a similar problem, their names are Watson and Crick, and they saw her, they were trying to figure out the structure of DNA too. They saw her picture, her supervising professor like showed them her picture without her knowing. And they were like, oh my gosh, that could work. We could figure out the structure for that. So within a couple weeks of seeing that picture, they figured out the correct way that all the different parts of the DNA lined up with each other. So they did this in 1953 and then would go on to win the Nobel Prize, which is kind of like the highest award you can achieve in science, I think in 1959, after other people had confirmed this is what the DNA looks like. So they made it out of these kind of like metal connector pieces and stuff like that to show all the different parts. But again, it has this sort of spiral staircase double helix look to it. So that's sort of the history of how we discovered what DNA looks like. But what is it actually? What are these different letters? Why do we keep calling it DNA? Don't those stand for something? They do, and it's a mouthful. So DNA stands for deoxyribonucleic acid, DNA. So the name comes from the fact that it's a type of molecule called a nucleic acid that has a special sugar in it called deoxyribose, which is where that D part comes from. So the Na stands for nucleic acid and the D stands for deoxyribo. Obviously, saying deoxyribonucleic acid every time that I wanted to talk about deoxyribonucleic acid gets very tiring. So everybody just calls it DNA, much easier. So again, we have these two different strands that are lined up with each other, but kind of spiral around one another, giving us this kind of crossing pattern as they go. So it's this long double-stranded piece that's twisted around, and then usually when it's inside your cell, it's really condensed down. So it's not like these loose strings that are floating around. It's all packaged together, mostly to save space, because there is a lot of DNA in your cells. So the Na part, the nucleic acid part, tells us a little bit about DNA. Specifically, that it is there to carry genetic information. So the information that makes you, you, the instructions in your body that make me, Michael, and make Kevin, Kevin, and that make Nasima, Nasima. All of that information is stored in your DNA. And next week, we're going to talk about exactly how we get that information out and turn it into stuff that's used in your body. But the instructions for how you developed when you were a baby inside of your mom, how you grew after you were born, the color hair that you have, the color eyes that you have, if you're a boy or a girl, if you have a certain disease or something like lactose intolerance or diabetes. A lot of that information is stored within the DNA in your body, the genes that make up your body. And this is information that you inherit from your parents. So your DNA is a mixture of 50% of your mom's DNA and 50% of your dad's DNA, all mixed together and turned into your DNA. So, like I was saying earlier, sure, DNA is this big, long structure that's all twisted around, but there are actually different pieces that go into building a single unit of DNA. So, DNA is something called a polymer, which is basically just a fancy science way of saying we have really similar pieces that are just copied over and over and over and over and over again. And so it's sort of like a big structure made out of Legos, right? If you see it from far away, you're like, oh, that's a cool pirate ship. And then when you look closer, you can see, oh, there are a whole bunch of little bricks that make it up. So our DNA, or another type of nucleic acid called RNA, that I'll talk about later on in the term, is made up of hundreds and thousands or millions of little pieces called nucleotides. So nucleotides are what are called monomers, which is like basic unit. So mono means like one or like singular, and it's like a prefix for that. So monomers are little individual pieces in DNA. They're called nucleotides. And if you put thousands or millions of nucleotides together, you could get one strand of DNA. So there are a couple different sections of a nucleotide, but we put tons and tons of them together, and we get two strings of them that wrap around each other, and that is what makes up our DNA. So these nucleotides have different parts of them as well. There is a phosphate group, which is this guy over here, represented by this little circle. We have a pentose sugar. So pentose is like pentagrams, like five-sided. And so we have this five-sided sugar, which is why it has this shape like this. And then finally, we have what's called a nitrogenous base. So in our DNA molecule, every single phosphate and every single sugar looks exactly the same. They're just copy pasted all the way up and down the DNA. The base, though, can be different. So I'll talk about them more in a second. But basically, there's 4 different bases that we can have: A, T, G, and C. So these guys can all be a little bit different from each other. There's just a slightly different structure that they come in based on what type of base that they are. But the sugar, the five-sided sugar, and the phosphate group are always going to be the same. So this is kind of what they look like. You don't need to know anything about the chemical structure. I'm just showing you. The phosphate group is called that because it is a phosphate atom surrounded by some oxygens. And then we have our 5-carbon sugar. So there's 5 carbon atoms that make up our little 5-sided sugar. And then we have our base, which would be one of 4 different possibilities. So phosphate, sugar, base. Every single nucleotide is made out of these 3 parts. Again, the phosphate and the sugar are identical for all the nucleotides, but the base can be different. And if we take this guy, connect it to a million of his friends, and make 2 different strands that connect together, you've got yourself a DNA molecule. So nucleotide is like our building block. It's like a little individual unit. And then the DNA is the big structure that we're able to make out of it. So just as kind of like a little aside, but the reason why we have the D in DNA, the deoxyribose part, is that that sugar, that five-sided sugar that goes into a nucleotide, it has a name. It's called 2' deoxyribose. So the reason why it has such a funny name is because in that other type of nucleic acid that I mentioned earlier, the RNA, we have what's called a ribose sugar. So you'll see these guys look pretty much identical, right? We've got this ring of carbons, these C's right here, all around. And we've got this O on top. And we've got this OH, CH2 guy up at the top. The only difference between them is that this CH attaches to an OH. And this CH just attaches to an H. So this guy is essentially missing an oxygen. So it's been, one of its oxygens has been taken away. It's been deoxygenated. So that's where the D in DNA comes from, is because the sugar doesn't have an oxygen compared to its kind of more standard cousin. So it's a ribose sugar that's missing an oxygen. So they call it deoxyribose, as in deoxyribose DNA or deoxyribonucleic acid. So that's just a little fun thing about those guys. So our bases right here, we have 4 different what are called nitrogenous bases, which is just a fancy way of saying that they're based on the element nitrogen. We have adenine, which is normally represented by A, guanine, represented by a G, cytosine, represented by a C, and thymine, represented by a T. You don't have to know anything about their structures, so don't worry about that. But they're pictured here. So adenine and guanine are like these double-ringed guys. Cytosine and thymine are little single rings. Excuse me. So again, this is the only part of the DNA that can be different. So in a DNA strand, usually going up and down the sides is where your phosphates and sugars are. So you have this phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, all kind of connected going up and down the sides of your DNA. A lot of times it's called like the sugar backbone of your DNA. And then meeting up across the middle, when you have your 2 strands next to each other, across the middle is where you get your bases. So these guys are the only part of the nucleotide that can be different from one another. And so again, this would be like a very, very tiny section of a part of DNA. Put another side over here and extend it out a couple million nucleotides, and you've got yourself a full set of DNA. Okay, so here's a picture that illustrates it a little bit better. So you can see we're connected through these little phosphates on either side, and those are connected to the sugar. So we got phosphate sugar, phosphate sugar, phosphate sugar running up and down the first side, and phosphate sugar, phosphate sugar, phosphate sugar running up and down the second side. And then across the middle are where our bases are. So our A's, our C's, our T's, our G's, they not line up kind of across the middle of the DNA. And they are what keep the DNA together. So this is why we're able to have the 2 different strands that kind of run right next to each other, because we have these bases going across and connecting to each other in the middle. All right. Before we go any further, Does anybody have any questions about DNA or the different parts of a nucleotide or the bases so far?
No, thank you, Mr. Michael.
No worries.
I'm good.
Cool, thanks guys. All right. So one thing that I want you guys to notice about our DNA here is that our two strands are not set up in the exact same way, right? If you look on the left-hand side, all the sugars are kind of pointed up, like the point is at the top part of the sugar and they're facing up. And then all the sugars on the other side are flipped around and pointing down. So this is something that'll come into play more next week, but I just wanted to kind of introduce it to you guys right now, is that the two different strands of DNA run kind of what's called anti-parallel to each other. So they're not like mirror images. If one is going up, then the other one is kind of going down. And so that's why you see the sugars and the phosphates in a little bit different orientations here. The other thing that I want you guys to notice is how the bases are lining up across the middle of our DNA here. Does anybody see any patterns in terms of how these bases line up together?
Yes, Mr. Michael. When we have the A, we have like T, we have G and C, and they go on like.
Yeah, exactly. So yeah, look at our little bases here. So the first one, we have an A that's connected to a T. And the second one, we have a G that's connected to a C. Okay, so A and T, G and C. Third one, we have a T that's connected to an A. And then an A that's connected to a T. Okay, so T and A seem to be really going together. And then C that's connected to a G, and a G that's connected to a C. So it seems like the A's and the T's tend to like to line up across from each other. And the G's and the C's also like to line up across from each other. And this is probably the most important thing about DNA that we're going to talk about today. And it is called the base pair rule. And it is a rule that said adenine and thymine, or thymine and adenine, will always, always, always line up together in a molecule of DNA. Excuse me. I had some allergy nastiness creep up on me over the weekend, and I'm for the most part feeling better now, but I still have like that one tiny little bit that's lingering over.
Hope you feel better, Mr. Michael.
Thank you. Yeah, almost all the way back to normal. Hopefully we'll be by tomorrow. All right, so adenine and thymine, thymine and adenine, ATTA, will always, always, always line up across from each other in DNA. Same thing with G and C, guanine and cytosine, or cytosine and guanine. They also will always, always line up to each other across side of DNA. So that's why we have on our little picture over here, we have an adenine on one side, which means we have a thymine on the other side. We have a thymine on one side, so we have to have an adenine on the other side. T on one side, A on the other. If we have a C on one side, we have to have a G on the other side. If we have a G on one side, we have to have a C on the other side. So again, we have our sugars and phosphates that are running up and down kind of the outside of the DNA, making that backbone. And inside we have A and T, T and A, always, always lining up with each other. And then we also have G's and C's that always line up with each other. So when we're talking about DNA, this will always be the rule, always be the case. You can always figure out what one side of the DNA is supposed to look like if you know what the other side looks like. Later on, like probably not tomorrow or Thursday, but next week we'll talk about RNA, which has a little bit different rules for how stuff pairs up. But when we're talking about DNA, always, A goes to T, G goes to C, C goes to G, T goes to A. There's never any exceptions. There's never any like breaks to the rule when we're talking about DNA. It'll always line up in this way. If they don't, if something happens and something gets switched around and they don't line up like this, that means that your DNA has a problem in it. And luckily, your cells are really good at finding that DNA and fixing it before that problem causes any issues. But they spend a lot of time checking your DNA and making sure that the A's and the T's and the G's and the C's are always lining up in the right way, in the right direction. So now that we know that rule, I want you guys to help me out here a little bit. So I've given you kind of different sets of one side of DNA at a time. And I want you guys to tell me, what should the other side of this DNA strand look like? So for example, if one side of my DNA reads ATA, what should the other side of my DNA look like?
T.
Good stuff.
So T and A.
And then what?
So TA and T.
Exactly. So if one side of our DNA is ATA, the other side has to be TAT. And A on one side means we have to have T on the other side. T on one side means we have to have A on the other side. So A and T will always go together. What about our next set of base pairs? Can somebody other than Nasima tell me if we have TCA on one side of our DNA, what should we find on the other side?
ATA. No, the same thing at the top, but flipped. So it'll be
Close.
CA.
Close. So you're right with the first one. So if you have a T on one side, then yeah, we should have an A on the other side. What does C pair up with?
C pairs up with A.
To G. So remember, A and T always go together. And so the other two that are left over are C and G. So a C will always pair up with a G. So our second set of three here, if we have TCA, that means on the other side, we have to have AGT. If we have a T on one side, there has to be a matching A on the other side. If we have a C on one side, there has to be a matching G on the other side. And finally, if we have an A on one side, we have to have a matching T on the other side. So A and T will always go together, AT, TA, and then C and G will always go together, CG or GC. All right, good start so far. What about our next set of 3? If we've got T, G, C on one side, what do you think we're going to have on the other side?
E.
Whoop, hold on, whoop, spoiling it. So yeah, I don't know if you guys heard him, but Kevin, who's in class here with me, just mentioned it. TGC is going to be ACG. So if there's a T on one side, we got an A on the other side. If there's a G on one side, there has to be a C on the other side. And if there's a C on one side, there has to be a G on the other. Yeah, Junior, question?
Yes, but who is the mean is the another size. So is that for the similar, for example?
Right. I think so. You cut out a little bit there. Can you repeat what you were asking again?
I mean, you see, is another size. This is something similar because is something difficult to understand who is the another size.
Yeah, so the sides will look similar since there's only the 4 different bases that you can have. But based on this pairing rule, you should be able to figure out what one side looks like based on what you have set up on the other side. If that's kind of what you were getting at. So like for our final group right here, we have GGC. And so what will that turn into on our next side?
G.
What was that, Naya?
It'll be CCG.
Yeah.
CCG, yes.
Mm-hmm. Exactly. Yeah. So G on one side means it matches up with C. G matches up with C. And C matches up with G. So based on what you have on one side of your DNA, these rules tell us exactly what should match with it on the other side of the DNA. So if you have an A on one side, it has to be matched up with a T on the other side of your DNA. If you have a C on one side, it has to be matched up with a G on the other side of your DNA. And same thing, if you go the other way, a T has to be matched with an A, and a G has to be matched with a C. So if we go back a couple slides here, so that's what we have going on here, right? We have our 2 sides of our DNA. A goes with T, T goes with A goes with T. And then also G goes with C, C goes with G, and G goes with C. So even if I covered up this side of this little strand here and you weren't able to see it, you should be able to pretty easily figure out what's supposed to be there based on what we have on our other strand over here. So that's what makes it the base pair rule. We call them base pairs because there are 2 of them, which makes a pair, and they're nitrogenous bases. So we have this AT pair, this GC pair, this TA pair, this AT pair. So these pairs of bases going down the middle of the DNA strands are called base pairs.
Mr. Michael.
Yeah, Nasima.
They're gonna be like always on the same shape, right? They never change this structure.
Mm-hmm.
Okay.
Well, when we're talking about DNA, it'll always be A's with T's, T's with A's, and it will always be G's with C's, C's with G's. Always, always, always in DNA.
Okay, thank you.
Like I said, next week, we're going to talk a little bit about RNA, which has a little bit different rules. It swaps out the T for a different letter, a U. But we'll worry about that when we get to it. Just remember, if you're dealing with DNA, this will always, always be true. No matter what one side is, you always have to have your A's and your T's match across from each other. And you always have to have your G's and your C's match across from each other. So in DNA, there's no exceptions. There's no exclusions. This will always be how the DNA lines up. So we have our sugar and phosphate backbone, which is represented in this picture by just these kind of solid blue lines. And then these base pairs, they're kind of sort of like the rungs of a ladder. So on a ladder, you have the two different sides going up, and then all the little steps going in between. And DNA, the base pairs are kind of like the steps. They're what connect one side of the DNA to the other, and they always pair up in this specific order. A's and T's, C's and G's. All right. Any other questions about DNA or the base pairs so far? Anything that anybody wants me to go back and explain about how the bases pair up with each other? Right. So there's only really a couple more things that I want to make sure to cover with you guys about DNA before we wrap up for tonight. So the way that the DNA is actually physically stuck together with those bases are things called hydrogen bonds. You don't have to know exactly how hydrogen bonds work. It essentially just has to do with the hydrogens on one of the bases are attracted to some of the atoms on the other bases. There's really only two things that you have to know about hydrogen bonds. One, they are what connect the bases to each other. So when you have those A and that T next to each other, the way they link up is through these bonds between them. So the hydrogens make kind of a weak attraction to the other one. The second thing to know about hydrogen bonds is that usually they're thought of as very weak bonds. So in other molecules and other particles that have hydrogen bonds, it's very easy to break them apart. The reason that DNA doesn't just fall apart and drift apart from each other is that there's so many bases in a single molecule of DNA. There's hundreds and thousands and millions of bases. So that is a lot of hydrogen bonds all connecting the different sides of the DNA together. So this is what actually holds our two sides of the DNA, holds those bases together. The T's and A's, the G's and C's, they're connected by these hydrogen bonds. Again, normally they're very weak bonds, but there's so many of them that it helps to give a really overall strong connection between the two. So that is what the little lines in this picture are supposed to represent. So the A and the T are not physically bonded and stuck together, but they're close enough to where those hydrogens are kind of pulling them into each other. And there's enough of those bonds across the entire big DNA molecule that the DNA is able to hold its shape. All right. So I mentioned that your DNA is really, really important because it has the instructions in order to make you. So these combination of A's, T's, G's, and C's, and the way that they specifically line up in certain genes determines what kind of traits that you're going to be born with. So imagine that we have this like weird alien cat creature thing. It also has DNA in its cells. And let's pretend that on the segment of DNA that determines what your hair color is going to be, this alien has the sequence CATCAT on one of its DNA strands. So this code will tell all the proteins and the hairs that go into building your hair color to make purple hair. So if you have CATCAT in your hair color segment of your DNA, your hair color gene, you get purple hair. But if that's different, if that's switched around a little bit and you get TACTAC, then you have yellow hair. So depending on the order that those bases come in, that is what determines the different traits in your body. And again, next week, we're going to talk about exactly how that works. But for now, just think of it as this segment of DNA controls a trait, and the order that the bases come in, the different combinations that they come in, determine what kind of traits that you're going to get. And while it seems kind of crazy that 4 letters determine everything about how your body works, I kind of like to think of it like the letters of a language. So, for example, the human English language is made up of 26 different letters, A through Z, right? Using those 26 letters, we can form hundreds of thousands of different words, right? There's a whole big book called the dictionary that lists all of the different words that we can form with the 26 English letters. And that's just in one language. We can then use the different words to form sentences, right? So even though we have all these hundreds of thousands of words, we could then have millions or billions of different sentences, all just based on ordering these letters in a certain way. So there's endless combinations of words and sentences and paragraphs and papers and books and stuff that could be written just based on the 26 letters that make up the English alphabet. Same kind of thing with DNA. Sure, there might only be 4 different bases, 4 different letters that go into building your DNA strands, but there are so many different ways that these can be rearranged in so many different orders that these can be put into, that's what gives us all the amount of sort of diversity of different people and different animals that we see on Earth, right? Every single living thing on planet Earth is made from the instructions in DNA. So whether or not you're like a bacteria or a mushroom or a strawberry plant or a dog or elephant or human, all of the information to make each one of those organisms is coded in its DNA. So everything on Earth that's alive uses these 4 letters in order to determine what sort of cells it has, what sort of body structure and body shape it's going to have, what are the different processes that can happen inside of the cells. That's all based on DNA that's been built out of these 4 bases right here. So we'll use these kind of bases as a starting point when we talk about on Thursday how DNA copies itself, because every single cell in your body contains a full set of your DNA. So every single cell in my body, whether it's a skin cell or a heart cell or a brain cell or a bone cell has all of Mr. Stanley's DNA in it. Every single one of your billions and billions of cells has a full set of DNA. It's not like the eye cells just get the eyeball part of the DNA and your skin cells just get the skin part. Every cell gets every part. They don't use every part, but they get every part. And so on Thursday, we'll talk about how it is whenever we want to make new skin cells or new heart cells, how we copy the DNA to make sure that it can go into the new cells that we're making. And then next week on Tuesday and Thursday, we'll talk about how do we take this DNA code and read it and turn it into the different structures and proteins in our cells that make it so that we can have all these different traits and all these different abilities. So DNA is really, really cool and fascinating. It's super important for all life on Earth. And so that's why we focus on it for the first couple weeks of biology here, because it does so many different things in our bodies. And it's so important, not just for us and not just for the animals that we live with, but literally every single living thing on planet Earth is based on DNA. All right. So does anybody have any more questions about the structure of DNA, the bases in DNA, the hydrogen bonds that hold it all together? Any final questions or anything that we went over today that you still want a little bit of instruction on?
Yes, Mr. Michael, please, like the hydrogen bond.
Sure.
If you can. Thank you so much.
So the hydrogen bonds are like the attractive force between the bases. So like A and T go together and C and G go together, but in the actual DNA itself, they're not physically connected to each other. Instead, they're just kind of attracted to each other with these hydrogen bonds. It's kind of like if you have a magnet and you hold it sort of close to another magnet, you can feel that they're attracted to each other. But in the case of these bases, they don't actually physically touch and come together. That attraction from one side to the other is due to these hydrogen bonds. And that attraction is what holds the entire DNA molecule together. Normally, hydrogen bonds, that sort of attractive force, are not very strong, and it would be very easy to break it apart and have the DNA flop around. But there's so many bases, there's so many millions of bases, all with these hydrogen bonds that are attracting them to each other, that it helps keep the DNA in the right structure, in the right shape. So it doesn't just fly apart because there's so many bases up and down the line holding it together. That's kind of what the little lines in our diagram up here are. The hydrogen bonds are attracting the different sides of the DNA to each other. So they're not physically like connected and locked in with each other, but they're close enough and they're attracted enough to where they're able to hold that DNA shape all throughout the millions and millions of base pairs that we have in the middle there.
But why, Mr. Michael, we have here, like when we have A and T, we have only 2 bonds, and when we have C and G, we have like 3?
That just has to do with the structure of the actual A and T molecules and G and C molecules. So it has to do with like the atoms that go into actually building these things. So like, I believe this is a G and this is a C. So just the way that they're set up, when they come close together, there's 3 different areas where the hydrogen bonds have that attraction. However, when you have an A and a T that are close to each other, the way the atoms are set up, there's only 2 areas that have that hydrogen bond attraction.
Okay.
So essentially, C and G have 3 hydrogen bonds between them, and A and T only have 2 hydrogen bonds between them. Again, you don't really need to know it. It's not super important. It's just kind of how things end up due to the actual like atomic structure of C and G, which is a little bit different than A and T.
And also, we don't have to know those words like tac tac to have purple hair. And we don't have to know those stuff. No?
Yeah. Nope, that was just kind of a really, really basic example of how DNA works.
Okay, thank you.
Mm-hmm. So yeah, this was just something that I made up. It's not real for anything that we know of, at least. All right, good questions though. Anybody have any other questions on hydrogen bonding, base pairs, nucleotides, or DNA? All righty. So since we are just kind of getting into the start of biology here, this might be for some of you the first time that you've really delved into DNA. I wanted to make sure we just covered the basics and the homework. So there are a couple simple little homework assignments for you guys to do. For today's homework, it's a really simple nucleotide labeling assignment where I have provided you with a basic nucleotide structure. And all I want you to do is label which one of these is pointing to the base, which one is pointing to the phosphate group, and which one is pointing to the sugar. So A, B, and C are all the three different parts of a nucleotide. And I want you to label which part that they're pointing to in the little labels down here. So that's for today's assignment. Again, should only take maybe a couple minutes, especially after going through the notes today. And then tomorrow's assignment has to do with these base pairs, so the ATCG stuff. And it's really a simple assignment, one question basically, although there are lots of little parts. So you have this one side of a DNA strand, ATG, CAC, TGG, ATC. And all I want you to do is tell me what would the complementary strand, which is like the technical scientific term for the matching strand, look like for this DNA strand. So this will be the homework that's posted tomorrow. And all you have to do is tell me, based on this strand and based on your knowledge of how the bases are supposed to pair up, what would the strand on the opposite side of the DNA look like? All right, and that's it. That's the homework for today and for tomorrow. And then, like I said, on Thursday, when we come back together, we'll talk about the slightly more complicated process of DNA replication, or how we take a DNA strand and copy it so that we can have new DNA for the new cells that we are building.
Are you gonna post this our homework for tomorrow on the thing?
Mm-hmm. Yeah, so tomorrow's homework will be posted at midnight, essentially tomorrow morning, and then it will be up for or available for the entire day. So you have all day tomorrow to work on it.
Okay, thank you.
Mm-hmm. Yeah, it'll go up in just over 6 hours from now, at the very start of Wednesday. All righty. There we go.